Some of our time.
No, now it is. All right. Good afternoon to those of you in the room and those of you on the Zoom. Welcome to the 11th Annual Wisconsin Science Festival and our conversation about fungi in Wisconsin, the good, the bad, and the ugly. I'm Laura Heisler, director and co-founder of the festival, and I have just a few housekeeping remarks and how-tos before I turn things over to our host, Larry Meiller. To get started, I want to, there we go. I want to start by thanking all of the amazing sponsors who make the Wisconsin Science Festival possible. Without their support, we wouldn't be able to bring you this fantastic programming free of charge. We do have hundreds more events yet today and tomorrow all across Wisconsin. So if you're interested in checking out more things, wisconsinsciencefest.org is the place to go. In terms of how we're going to proceed today, we are going to have folks on the Zoom and folks here in the room. We're going to have a great panel conversation, and then we're going to open things up for your questions with about 15 minutes before we end. We really do have to end at 2:45 because we have another event in here at 3. We have 2 more, if you're interested, 2 more Wisconsin Science Festival meets Wisconsin Book Festival talks. At 3 o'clock, we have Jordan Ellenberg on his book, UW-Madison's own Jordan Ellenberg with his book on shape. And at 4, we have Larry Olmsted with his book on the science of fandom called Fans, if you're interested in sticking around. So when we go to Q&A, we're going to ask for those in the room that you line up by the microphone here in the middle of the room. That's our COVID touch-free way, recommended touch-free way of fielding your questions. If you're on the Zoom, please use the Q&A function and I will be your spokesperson and we will try to toggle between questions in the line and questions on the Zoom. So that's how we're gonna go. And if there are a lot of questions, I apologize in advance. We may not get to all of them. With that, I am delighted to introduce Larry Meiller, professor in the Life Sciences Communication Department here at UW-Madison and longtime revered host of Wisconsin Public Radio as our moderator. And Larry will be introducing today's panel, some of whom are in the room, some of whom are in the Zoom. So with that, Larry, over to you.
Well, thank you, Laura. And I want to say hello to everyone. Thank you so much for being a part of this festival panel talk today. We have for you 5 experts on the fungi of Wisconsin. Worldwide, you know, we have 144,000 about known fungi species, and I'm told about 10,000 of those species reside in our state. And we're talking yeasts, rusts, smuts, mildews, molds, and my favorite, mushrooms. They make up the species of fungi, and they're amazing living things. A guest on my show told me that fungi produce worldwide 50 megatons of spores per year, and that would be the equivalent of 500,000 blue whales, the weight of the largest animals recorded on Earth. So here in Wisconsin every year, we get some tons of spores, and those spores produce more tons of fungi, and that's what we're going to be talking about today with our 5 guest experts. With us, we have Amanda Gevens from UW Plant Pathology. Raise your hand. Chris Hittinger from UW Genetics. We have Leslie Holland from UW Plant Pathology. Michelle Jusino from the U.S. Forest Service, she's waving, and Dan Lindner from the U.S. Forest Service as well. And I want to start with Michelle Jusino. Michelle is a research biologist with the Forest Service. She works out of the Northern Research Station based here in Madison. And she studies fungal communities that are important to maintaining forest health and ecosystem functions as well. Her research topics are ranging. They range from the structure and function of decay communities to the relationship between fungi and animals, birds, bats, and beetles. Michelle, I know there are good fungi for animals, and there are some bad ones as well. Let's talk about the good ones first. What are some ways that fungi can be good for animals?
Hi, thanks so much for that introduction. Larry, can everybody hear me okay?
Yes.
Okay, great. So I have some slides. If you could advance to the first one, please. So fungi can help animals in many ways. One way is by helping them accomplish things. So here you see this panel of pictures that may or may not look familiar to you. So this is in the longleaf pine ecosystem in the southeastern United States, so not in Wisconsin, but I'll tie it into how it relates to Wisconsin at the end of this story. And here we see the endangered red-cockaded woodpecker. So this is one example of many in which fungi can help cavity excavating and cavity nesting birds. So woodpeckers make holes, or most of them do, right, in trees. And decay fungi soften wood in trees. And sometimes this allows animals to make excavations or holes. And they use these holes to nest in or to roost or sleep in. And so this red-cockaded woodpecker, it takes a really long time to make a hole in a tree. Most excavators take weeks to months. These birds take years. And the birds need these holes, okay? Without these holes, they will die. Each bird needs a hole. They use it to sleep in every night. And fungi have been hypothesized to help these birds. So they would help them by quickening the excavation time to lessen it from something like 8 years to maybe something more like 2 years. So here you see a picture of a fungal conch, which is Portobellos pini. And this is a fungus that was long thought to help these birds. And you can also see, let me see, I'm not sharing my screen. You are sharing the screen, so I can't mouse over. The picture that looks like a golf ball that hit the tree in the middle of the screen there, that's an incomplete excavation. So you can see as these birds make their hole, they have these wounds on the tree, and they could be courts for fungi to come into the tree while the bird is excavating their cavity. And so we actually found that these birds are associated with not just Portobellos pini, right? This one decay fungus, and not just 2 decay fungi, but many decay fungi. And the birds actually help transmit the decay fungi from tree to tree. And the fungi may also be helping these birds by quickening their excavation time. So you can see a little bird making a hole in a tree, and that slide on the side of the screen, and then finally a complete cavity on the bottom. So we see relationships like this with other woodpeckers, with other cavity excavators that aren't woodpeckers too, including the black-capped chickadee. So we have black-capped chickadees in Wisconsin, and we've seen that relationship with black-capped chickadees in northern Wisconsin and also in the Upper Peninsula of Michigan. Okay, next slide, please. Another way animals can benefit from fungi is by eating them, right? We eat fungi, some of us do. Some people don't like to eat them. Here you see some more birds and a lot of different types of truffles. That's what these little fruiting bodies are. So most of us, when we think of truffles, we think of these gourmet fungi, right? Very odoriferous fungi that are dispersed by mammals. And maybe you think of truffles and you think of, like truffle hunting dogs and stuff like this. But here we have birds that are eating truffles, hunting them and eating them in South America. And so these two birds, these are a ground-dwelling bird family. Rhinocryptidae is the family. We have choucals and wetwets. This is in Patagonia. But anyways, so truffles are fungi that fruit below ground. Many of them have ectomycorrhizal relationships with trees too, right? So this is another kind of beneficial story nested within this story. And most truffles, like I said, are dispersed by mammals and are highly odoriferous, right? They have strong smells that we really detect and really like. These truffles don't have these strong smells. And so the question was, like here we see this great diversity of truffles, but what's dispersing them? What's transmitting the spores, right? And it's actually, we have evidence that these birds are doing that. And so those are two good stories. So I'm ready for the next question, Larry, and the next slide.
Yeah, the bad side.
Yeah, so this is kind of a mix. And I think I have time to tell this one, yes. So here you see ambrosia beetles and their galleries of fungus. So there are some animals that eat fungi, right? I just told you about this. There are some animals that eat fungi and are specialized so much that they farm fungi for their food. So these tiny little beetles, these are ambrosia beetles. Many of you have probably heard of bark and ambrosia beetles because sometimes their fungi can be very detrimental. And we have these in Wisconsin. If you go out in the woods in the spring or the fall, you'd be able to find some of these little beetles. They drill tiny holes in the tree. And as they go into the tree, they carry fungi with them in little fungus pockets, right? So these fungus pockets can be in their mouths or sometimes in the back, behind their head. As they go into the tree, they inoculate the wood with fungi, and then the fungus grows. You can see these nice white fluffy mats. So the beetle goes in, it lays its eggs, it makes these tunnels for its larvae. The larvae grow up and they eat this nice white fluffy fungal mat that you see. And these are all different types of fungi. Most of these fungi, I should say, and most of these beetles are not harmful, but there are very few that are, and this is why a lot of us know about these relationships. So usually they don't attack or kill healthy trees, but sometimes they do. Okay, I think I'm out of time.
Well, you're close, but so fungi can, they're good for animals, but they can sometimes be bad for us, right? The ones that are good for animals.
Yes, so some of these fungi that are associated with ambrosia beetles can really take out entire forests of trees. So there's, for instance, the red bay ambrosia beetle and its fungus that causes laurel wilt is a very big economic and agricultural issue. If you've heard of pine beetles can decimate forests. So that's one example, yes.
I've heard some almost swearing on my radio show when it comes to those beetles, as a matter of fact. And I'm sure there was plenty of swearing about it off the air from those people. Thank you, Michelle. Well, we can come back to you, but I want to turn to Amanda Gevens. Amanda chairs the UW-Madison Plant Pathology Department. She's a professor, of course, an integrated extension specialist as well, and she studies pathogens and their resulting diseases, especially as it relates to the production of potato, vegetable, and specialty crops here in Wisconsin, and of course beyond Wisconsin as well. About half of the pathogens she studies are fungal in nature, and she develops integrated disease management programs for these crops. And she also focuses on the ecology of plant pathogens within the whole system, the whole ecosystem of food production, storage, and transit. So she looks at all of those things. What makes, Amanda, a fungus? What makes a fungus a plant pathogen?
Yeah, that's a fine question, Larry. What makes a fungus a pathogen? Well, in our department, I'll start with this. In name, we already sort of filter plant microbe interactions in a very negative light in the way we name our department. plant pathology. But these fungi are pathogens because they make food source out of plants, right? And that's very simply put. But what pathogens do is they find a way to create a disease, first home on a plant and plant tissues, and then create a disease. And that's the requirement for pathology, right? The pathogen has to infect and then cause some kind of a malady on the plant. And that can be a spot, that can be a speck, that can be a blight, that could be wilting, and ultimately it could be plant death. So pathogens have to be able to infect and cause malady on the plant. We have many microbes that live in a community with plants. Those are endophytes. Those are beneficials that do good for the plant in its life. In fact, some fungi will help to limit the negative effects of pathogens in a plant's life. So more genuinely and truthfully in our discipline, we do study plant microbe interactions, but pathology is certainly the root of our work. And I think in there I've addressed your question.
Yeah, where do these pathogens hang out when they're, they have to wait around, I assume,
Yes.
for host plants or crops?
Yes. So where pathogens are hanging out, where they residually are living between cropping systems, it can vary depending upon the fungus. And so we do have some fungal pathogens of plants that have an overwintering structure. These things are named variably depending upon the type or the taxonomy of the particular fungus. But I should say most plant pathogens they're microscopic, right? So when we show pictures of fungi, and certainly some fungi that are, we've seen some lovely photos here behind me, but shelf fungi and bracket fungi, things on trees have a very pronounced phenotype, right? We can see those well. They're quite lovely. In plant pathology, a lot of the pathogens that we interact with and engage with are quite small. They require a microscope. They may be about 20 microns in size, and their spores are that size or smaller, and they get around aerially, and we don't necessarily see them. They move about like pollen does. And so they're microscopic, and some of these microscopic structures can reside in the soil, and they're what we call overwintering spores. There may be a larger fungal structure that is a mass of mycelial, or essentially the root structures of fungus, if you will, that can reside in the soil long term. And what makes them work is that they have the right conditions to make disease. And so what I'll introduce now is the disease triangle, which is a foundational principle in plant pathology. But that triangle at each corner has a critical element to make disease. One element is a susceptible host. And in the crops that I work with in Wisconsin, that's potatoes, or it's carrots, it's cabbage, sweet corn, step beans, beets. But that susceptible host is essential. And then we have to have the right environmental condition. And for most fungal pathogens, and fungi make up 70% of plant pathogens globally, fungi need moisture, right? So it's typically, there's some element of either free moisture, meaning there's a film of water on the plant, or it's high humidity, but we need moisture. And often it's moderate temperatures. And in Wisconsin, we sort of have those environmental conditions licked, right? We have springs that are quite wet and moderate. Our summers get hot, but often not far above 90, 92. So we have really good climate for fungal pathogens in plants here in the state. And then the final corner, the final component of this triangle is a virulent or a disease-causing pathogen. And as I mentioned, some of these can be hanging out in the soil waiting. Some of them can take a ride on air currents and come to our state from more southerly places where we see crop diseases more ample or routine given their climate and their cropping systems. And other pathogens will hang around in seed. You probably have heard of seed-borne pathogens. Those are problematic on both true seeded crops, things like carrots and beets and beans and corn, as well as crops that are propagated with plant parts like potatoes. These are seed potatoes that in and of themselves have a microbiome. And so when you plant those seed pieces, you're planting microbes, and some of those microbes might be pathogens of potato. So those are just some of the places that pathogens can hang out.
So what are, as you do your research and your extension work on this, what pathogen is giving you the biggest headache right now, or giving farmers, let's say in the state, probably the biggest headache right now?
Yeah, that's a good question. We work with a number of pathogens, both fungal and those that are fungal-like. What I'll speak to is one pathogen that it's quite common, and it probably costs our potato growers the most, both economic as well as headache and emotional distress. And that's a pathogen that's called Alternaria solani, and it causes early blight. And Alternaria, they're very common. This genus is quite common. There might be some Alternaria spores in here right now, and you just don't see it and know it. But Alternaria solani is it's an annual problem in potato and tomato crops here in Wisconsin. The pathogen resides in crop residue, and we have the geography of Wisconsin is such that we do have concentrated potato production in central Wisconsin, and we have a few other pockets of concentrated production around the state. But those areas are fine homes for Alternaria solani fungi to hang out in residue. And so we studied this particular disease in potatoes. We look at varietal resistance as an option. We look at crop rotations to try to reduce that residual inoculum in the environment. And we look at other strategies like use of biological controls, biopesticides, as well as other crop protectants for control of disease.
So that's a big headache. I hear that on my radio show, on my Garden Talk show on Fridays. That comes up pretty often. Well, we can come back to you, Amanda, but I want to go to Leslie because she works on a different kind of aspect. You work on vegetable related things, and Leslie's more likely to be working on fruit. She's an assistant professor and extension specialist at UW-Madison in the Department of Plant Pathology. And her research focuses on diagnosing and managing fruit crop diseases for our state's fruit industries, cranberries, apples, grapes, specialty fruit industries. And then she takes that extension program, that side of it, she translates the research findings into hopefully practical solutions to improve productivity and sustainability of our fruit crops. So Leslie, can a good fungus control a bad fungus?
That's a great question, and thank you for the introduction. Yes, they can, but maybe a question before that some of you might ask is why would we even need that, right? Oftentimes when we think about agriculture, we have things called fungicides, right, that kill fungi. So why would we need a good fungus to kill these bad plant pathogens like we've been talking about? Well, fungicides are effective and they are widely used, but sometimes either in organic agriculture or perhaps lower input systems, a lot of growers and even homeowners are interested in using products that are less harmful to the environment, but also just have an alternative mode of action. And that's what biocontrols can offer. And that's what we call them, as a biological control. So oftentimes a lot of biological controls are formulated with fungi, the good guys, as we like to call them. And so there's numerous fungi that can actually help manage bad fungi, so to speak. And so they do this in a multitude of ways. So in terms of how they control them, it could be competition. It could be simply the good fungus can grow much quicker than the bad fungus and compete for space. They can compete for nutrients too, perhaps, right? Whatever the plant, as Amanda mentioned before, is providing that pathogen, perhaps that biocontrol fungus can utilize that much quicker, much faster, or in a better manner than that pathogen is able to do so. There are some biocontrol fungi that can actually produce chemistries that either are inhibitory to the particular pathogen, or they can produce chemistries that actually kind of tap the plant on the shoulder and say, hey, you got to watch yourself. There's a pathogen here. So there's different modes of action that these good fungi can use in order to prevent these bad fungi from taking over. And in agriculture, we do have some products that are actually formulated from lab research that growers are actually able to use and apply to their cropping systems as a means of control. So while we do have a lot of fungicides in use, there's also a fair amount of biocontrols that are registered by different companies and are currently in use. And so those are always exciting to see. Now, thinking about the disease triangle that Amanda just introduced to everybody, there are some difficulties with that. The same way that plant pathogens need certain environments to survive and infect and do their thing, so to speak, so do biological control agents, right? That same humidity, those same environmental conditions that are conducive for fungal survival, those are the same things that are needed for those good fungi to survive and do their job as well. So that's something we have to keep in mind as scientists when we start to try out these biological control products. Is the environment in a good state for them in order to manage the bad fungus?
How do you insert those fungi into the system?
That's a great question. So oftentimes when we find a fungus that works for us, actually, I think shame on me for not showing my slide I put together for everyone today. I'm so excited to talk about it. So this is actually a really great example. So I'll answer Larry's question first. How do we integrate this into a system? Oftentimes we find a fungus that works. Trichoderma is a really excellent example of that. And that's this green one. Oops. It's the green fungus that we see here, right? And so this fungus, you can see it's a quick grower. It's really taking over the plate compared to this pathogen, which is botrytis on this plate. So it's seeming to really outgrow. So when we find a really great candidate like this, companies are able to formulate that into some type of granular or powder or sand or some type of formulation that can be tank mixed with the water, just like a fungicide would be suspended in tank mix with the water to spray on crops. So it's very similar formulations that we make. Oftentimes they need to be stored a little bit differently, right? Because you're now working with a biological organism, so that might need to be kept in the fridge. And it's probably not going to last on your shelf for too long, right? Because you've got this active ingredient in there. But yeah, typically formulated like that. And as I mentioned, trichoderma is a really excellent example of a biocontrol that has been widely used for control. So it's a nice green one here.
And can they, I guess Amanda said they can overwinter, but maybe talk a bit more about how they survive. I mean, we got a La Nina winter coming, the weather guy says. So how do they survive the winter?
Absolutely. Yeah. Wisconsin. I'm not from Wisconsin originally. In fact, I've survived my first Wisconsin winter. It was great, but it was a tad chilly. And so what it makes you think, how are fungi surviving these winters, right? We just talked about sometimes warm temperatures, moist conditions, right? That sounds great. It's a tropical environment almost that these fungi are thriving. But then winter hits and it's cold, it's potentially dry. How are fungi surviving in this? And Amanda did a great job of introducing a term we use called overwintering. And so fungi have really diverse ways of overwintering in different cropping systems. So I'm going to just mention a couple of those. Okay, so I've got some photos here of how fungi can overwinter in different. I give you a sneak preview for our next. I apologize. Those are some great photos, though, weren't they? So I can admire all fungi. I really can. So our top left corner here is an example of apple scab. So this is an issue that homeowners see, commercial growers see, where scabs are these little tufts of fungi grow on leaves and they can grow on apple fruits as well. And so we see this during the growing season. It's frustrating. We apply chemical control products and that usually manages it. But at the end of the season, where we are right now, those leaves fall off of the tree and they fall into the orchard floor, right? And we're not typically thinking about raking those up, not when you have 4, 5, 10, 15 acres of apple tree, right? Raking's not an efficient thing to do. So the leaves tend to stay there. And so does the fungi, which is living on those leaves. And so what happens is those fungi during the winter period, they can just live in that debris on the orchard floor undisturbed. And then come the following spring, when you're hoping your buds break and you're getting production of your apple fruits, under right conditions with humidity, rain, these fungal spores will shoot up again. So fungi find really great mechanisms to just survive in old tissue. That's really their secret, right? It's not particularly impressive in a lot of ways, but it's how it's done. So leaf tissue is one example of this. One of my favorite examples are canker fungi. I've had the opportunity to work on these in several cropping systems, and I find them fascinating. And oftentimes when you walk through downtown or wherever you live during the winter, there's not much to see. I'm going to challenge you now to look at different trees and ornamentals in your neighborhood and see if you can find fruiting bodies, right? We often think of fruiting bodies as these beautiful conch fungi on trees or fruiting bodies coming out of the ground, these mushrooms. But I challenge you to look for the tiny pinhole fruiting bodies coming out of twigs and branches on trees. And they look like this. Almost little pimple-like appearances. This is another really critical way that fungi are able to survive in the winter is by forming these different fruiting structures. And if you cut some open, sometimes they look like this, little locules, and that's where all the spores are. And they're gonna wait until a raindrop hits them and they can eject and shoot and affect fresh tissue in the spring when we get those spring rains. So these are just some examples of how fungi are able to overwinter. We have blueberry here in the middle. It's a little difficult to see, but these are apothecia or cup fungi that are forming out on top of the blueberry as well. So just different mechanisms that we see fungi surviving these cold winters in dead tissue.
I think that's pretty cool, although I'm surprised you haven't invented a giant vacuum cleaner to vacuum up the leaves in the orchards.
Yeah, we
You could
haven't.
call it Leslie's vacuum.
That would be an excellent name. So growers, fortunately, and researchers have actually found some ways for the instance of apple scab specifically that they're able to use certain chemistries or products to apply to the leaves on the ground before bud break to help actually kill those fungi, right? So you're killing them at the source, which is in the debris. So a lot of our cultural or even chemical practices used in agriculture are often targeted overwintering structures, right? We often think about targeting them at the lush, beautiful green tissue or fungi coming in from far, but oftentimes we're targeting them at the fungi that already exist on these dead tissues in orchards or in fields. So this is a really critical point for management for a lot of growers.
You know, and I see, you can see maybe fungi on plants and that could, on a fruit of some sort, and that, if I'm looking at one, I'm probably not going to eat that, but can fungi affect the taste of the fruits themselves?
That's a great question. Yes, they can. I don't know if you've ever bitten into a fruit that you maybe realized after the fact that probably had a little something funky going on with it that was fungal in nature, and it can, right? They're after the same sweetness and the sugars that you are as well. And so some of that can be potentially fermented or just broken down and become mushy or just have a kind of off taste to them. So they definitely can. I'd say one benefit of fungi though, and wine, if anybody drinks wine around here, botrytis, which I showed on the previous slide, has actually been used in winemaking to make these botrytized wines, right? It's a fungal pathogen that really can ravage vineyards, but somehow we found a way to say, hey, working with nature and making these botrytized wines that people really enjoy drinking. So
Who knew?
the good and the bad, right? So I snuck some good in there.
Well, let's turn to Dan Lindner next. Dan's a research mycologist with the U.S. Forest Service. He's based in the Forest Products Lab in Madison, and he studies fungi important for forest health, including both pathogenic and beneficial fungi. And for his PhD, I thought was pretty cool. He studied how forest management affected fungal communities in northern Wisconsin. And his research spans a range of topics from wood decay fungi to white nose syndrome in bats. So it's pretty broad if you ask me. Dan, are wood decay fungi considered good fungi? And can wood decay fungi be bad?
Yes. Well, I want to talk about wood decay fungi today because especially some from Wisconsin, because we tend to focus on pathogens and they're often what we think about because they're economically important. I'm actually trained as a plant pathologist. But one of the things that fungi do and that they're known for is decomposition. And it's this question of, is that a good thing? And I'd like to convince you that it is. It's something that we should think about, that it's critical for forest health and for soil formation. And there's just a couple of examples of fungi, decay fungi from Wisconsin here, and it actually leans towards ones that I love that happen to be edible fungi. The one in the upper left is the velvet stalk fungus. So sometimes they look more like traditional mushrooms with gills like we think, but they can have this variety of forms and colors and shapes. On the right, we've got chicken of the woods or sulfur shelf that has these almost candy corn-like colors, appropriate for this time of year, or even toothed fungus like this lion's mane in the lower left. So they come in a range, but actually, if we could go to the next slide, even more obscure, oh wait, one back. Before we get to that, I encourage people to turn over logs because we focus on the charismatic mega fungi, the big beautiful ones, but when you turn over a log, there are hundreds of species of wood decay fungi that sometimes just form crusts like this. This red species is a wood decay fungus. The white species is also, that's Hyphoderma puberum. The little cups in the upper left, that's an ascomycete that's also helping to decay the wood. And the Phlebia coccinea fulva, that's that beautiful red one, I think it's beautiful, with the yellow margins that we find actually in the driftless area of Wisconsin. We don't know why it tends to be, it's very common in the driftless area of Wisconsin. We don't find it in most of the other parts of Wisconsin, but it's considered rare enough in Europe, actually. Many in Scandinavia, different countries list it as on their rare or threatened and endangered species list. We don't have great lists of endangered species of fungi for North America because we often know so little about their distribution. But this shows you a range of decay fungi. But why are they important and what is it that they do? They're important because they're really the only organisms that efficiently can break down wood. Wood is an amazing substance. It's really strong. It's strong because it has something in it called lignin. And plants invented lignin about, say, 360 million years ago. Plants started to colonize land about 400 million years ago, but they were all very short. They were very small. We didn't have trees because we didn't have wood. There was no wood. Plants started to get the enzymes to synthesize lignin, and all of a sudden you could make wood and trees could become tall and there were forests. But their wood came on the scene kind of like plastic. It was this amazing substance, really hard to break down. And in fact, there were no microorganisms at the time that could break it down. And there was about a 60 million year gap between when wood came on the scene in Earth's history and when fungi finally cracked the code and figured out how to start to break down wood. And that 60 million year period actually coincides with what we call the Carboniferous period, when many of the coal deposits were formed, where we had this huge carbon sequestration event because wood was piling up in forests hundreds of feet, thousands of feet deep, and not decomposing, but then fossilizing and actually turning into coal. Essentially, fossilized lignin is what we burn as coal today. And you still find lots of plant fossils within coal. So fungi cracked the code and kind of switched the earth. The theory is from carbon sequestration, massive carbon sequestration, to carbon cycling, what we know today. And that's one of the things that, from a research standpoint, that I'm very interested in terms of forest management, of how we can work with forest management techniques that encourage decomposition and getting decay fungi to move carbon into the soil for the long term for forest health, rather than particular situations where you can wind up with fungi that put almost all of that carbon very quickly back into the atmosphere rather than storing it for longer periods of time in the soil. And in terms of how they can be bad, potentially, if we could advance to the next slide. I hate to ever call a fungus bad. Intrinsically, they're not bad, but if you're from Wisconsin, you may have noticed an explosion of fungi that are kind of out of place. Golden oyster mushrooms. It's an invasive species that it's native to Eastern Russia. It's grown by growers in many places, and it has escaped cultivation. And especially if you're out, say, morel hunting and looking at dead elms, you'll often see that these elm trees are just massively colonized by golden oysters. And in this case, these are decay fungi, but they could be bad because one, they're so good at doing their job. They may be displacing a lot of our native oyster mushrooms and other decay fungi. And they may take a log that could slowly store carbon and decompose over 20 years. And these oysters are bred to chew through trees. And what I've seen just anecdotally is that often in 2 years, that instead of decomposing over 20 years, when the tree gets hit by these golden oysters, it looks like there's practically nothing left of the tree 2 years later. People still love to see them because it's an edible fungus. They're beautiful. But this is part of our goal to try and think about that there are invasive species of wood decay fungi too, and that try and come up with strategies so that growers have resources for go local, grow local, get our local strains of wood decay fungi, and try and have those be commercialized. But it just hasn't been done yet. And that's one of the things that we're really excited to try and get more local strains so that local growers, small mushroom growers, can be producing things that don't come from Australia, or we pick them often because they're beautiful in color and shape and form. But we have lots of beautiful fungi from Wisconsin that we can also grow.
Dan, thank you. And we can come back to you with questions later. And I'm sure there may be some questions from our audience as well, but I want to get Chris Hittinger involved. Chris is an associate professor of genetics, the H.L.I. Romney's faculty fellow, and the primary fuel system synthesis. I've had trouble with that word my whole life. Synthesis team lead of the DOE Great Lakes Bioenergy Research Center. His lab studies the biodiversity, studies biodiversity, evolutionary genomics, and carbon metabolism of budding yeasts, including applications in beer and bioenergy. Dan, yeast. What are yeasts and how are they related to other fungi?
Yeah, thanks for that introduction, Larry. Yeasts are single-celled fungi that don't form the beautiful fruiting bodies that Leslie and Dan have introduced us to. And so the way that they're related to other fungi is that these single-celled yeasts have actually evolved multiple times from multicellular fungal ancestors. If we can go to the first slide here, the largest group of yeasts are the budding yeasts, and these evolved about 400 million years ago, probably from a mold-like ancestor. And as you can see, the axis here is showing genetic divergence. And what you can see is there is as much genetic diversity in these budding yeasts that don't actually look that different from one another as there is in the entire animal kingdom, and really more than you see in the entire plant kingdom. So despite the fact that there's not maybe a lot going on morphologically, there's really a lot going on under the hood, as it were. And so despite those similar appearances, they differ substantially in what they like to eat. And so in particular, they like to eat different types of sugars or other carbon sources. And they also make different types of carbon products. They make different decisions about what to do with that food as they eat it. And that's a kind of a major way that they subdivide their ecological niches in nature.
I was going to ask you where yeasts are found, but I think one of the common places is probably in the cupboards in the kitchens of everybody who's here today. I went and looked to make sure I still had some yeast packets in my cupboard this morning. But where are they found in nature, and what are they good for? We know they're good for bread and beer too.
Yeah, sure. And of course, there was a great shortage of yeasts. I know during the early days of the pandemic, as people were hoarding. But yeasts are actually found everywhere, though. They're found on every continent, even Antarctica, and every ecosystem, whether they're terrestrial or aquatic. And so you can find them on fruits, in the soil, bark, insects. They're even inside all of us. But since they're single-celled, we just can't see them in nature because they're too tiny to see without a microscope. They're also generally, in most cases, pretty low abundance members of communities, and so they're not often a significant part of the microbial community. But what we can do is we can enrich for them and isolate them in the lab. And so if we go to the next slide, this is actually one of the major ways that we train our new undergraduate students in the lab, is we just send them out in the field and tell them to bring back soil or fruit or leaves and actually enrich for them in the lab. And so we just take whatever sample that they bring and the students make a sugar-rich media where they add antibiotics to kill off the bacteria. And we also limit the oxygen to give the yeasts an edge over the filamentous fungi that you've been hearing about from the other speakers. And if you go through that a few times, you can then put them on the plates. And then the one yeast cell will grow up into millions of cells, and that's called a colony. And we can actually pick that with a toothpick. And then we do various genetic and molecular procedures on them and bioinformatic procedures on them to identify them. And so we call this the Wild Yeast Program, the Yeast Exploration and Analysis Science Team. And this is really how the vast majority of undergraduates in our lab have been trained, is by isolating, what are now thousands of wild yeasts, mostly from Wisconsin. So we don't, most of the yeast that we isolate, actually, we don't know much about them. They've maybe been isolated once or twice by other people around the world. In some cases, there are new species, and we've already described more than a dozen new species, and there are dozens more whose descriptions we're working on. But some of the yeasts, though, that you probably do know of, we do also isolate from nature, and they are responsible for some of the yeast products that you encounter every day. And most of these have to do with fermentation. So if we go to the slide, we can see sort of a picture of a cornucopia of yeast products on the next slide. And so these are examples where yeasts are generally fermenting available sugars. And so Larry, I think, alluded to the great yeast shortage as everyone was home baking. And so bread is a major yeast product. And so what the yeasts are doing there are actually fermenting the sugars that are inside the dough and helping it rise by producing the carbon dioxide. Can also take those same types of sugars and ferment them into alcoholic beverages or mini fermented food products. And so there you're not only taking advantage of the carbon dioxide that's produced, but also the ethanol that's produced by fermentation. There's also esters and other products that are actually imparted that impart unique flavors to fermented foods and beverages. And this is also used in the cheese industry. There are a number of yeasts that are either deliberately inoculated or passively accumulated from the environment that actually are used in cheese ripening and impart unique flavors to cheese. Of course, some people will actually eat yeast itself. So you can have Marmite or Vegemite are sort of the trade names. And some people actually will take yeast as a probiotic. And then, of course, we're now increasingly looking to use yeasts not just for the kinds of agricultural products that humans have made for thousands of years, but actually looking to yeasts to help us build a sustainable bioeconomy that's carbon neutral. And that's really important because yeasts are such great fermenters that we can use them to make fuels and chemicals from plant biomass that does not compete with crops. And that's some of the work that we do within the DOE Great Lakes Bioenergy Research Center, is trying to learn how to reprogram yeasts to make advanced biofuels for us.
It's pretty cool. As a matter of fact, I think, and another good thing, and I think you're connected with this. Are you involved in helping the students brew beer on campus with a brewing class?
Yeah, so we have some collaborators over in food science. Monica Thies and Nick Schweitzer are the main ones involved with that, but they have used some of our strains to brew with. And so that in particular, there's been a lot of interest brewing with a species that we discovered about a decade ago that's called Saccharomyces eubayanus. And it turned out to be the wild ancestor of hybrid lager brewing yeast. And so it's a cold tolerant yeast that actually hybridized with domesticated ale brewing yeasts. Saccharomyces cerevisiae is the most famous yeast, and it's used to make most wine, and it was traditionally used to make ale beer. But when people started brewing at colder temperatures, Saccharomyces cerevisiae is not happy. And so this cold tolerant species came from the wild and actually formed an interspecies hybrid. And that actually kicked off the lager brewing industry about 500 years ago. And that, of course, constitutes the majority of the global market now. And so they've been trying to figure out how to domesticate that wild species in sort of a non-hybrid context. And working on producing interesting beers with it.
So to our audience, there you have it, an introduction to all 5 of our guest experts and what they're involved in. They know a lot more than what we've talked about in this brief time, but I'm hoping that you have some questions for them. And there's, I believe there's emails, but you can also come right to the front and I'm sorry, to the microphone and ask your questions. So, and we're getting some folks coming to the front. So
Can I
please
ask
go
one
ahead.
from online before we jump in? And I'll just
Oh.
also mention, we do have to end in 10 minutes. So just to keep your questions succinct. Dan, you answered this question online, but I'm guessing lots of folks in the audience are wondering as well. What about those golden oyster mushrooms? Can people just go out and harvest them and eat them?
If you know what they are, so golden oyster mushrooms are edible. They're delicious. That's why commercial growers were growing them. But it all comes down to knowing your mushroom identification. As they always say, every mushroom is edible once, but you hope to be able to eat it more times than that. So I recommend joining the Wisconsin Mycological Society. It's hard to identify mushrooms from books. The way I learned it, now these days, everyone wants to try and do it with an app on their phone. Get to know people, get to know the people, the experts. We've got plenty of them in Madison, wonderful amateur experts. And through the Wisconsin Mycological Society, they go out on forays and they can teach you. So if you're not confident about identifying your oyster mushrooms, hook up with the Wisconsin Mycological Society, go out on forays, learn your mushroom identification, and don't eat anything that you're not 100% sure of.
Amen. Please go ahead.
This might be a question for Dan. Yesterday, there was a great talk here on the global soil erosion crisis in our farms and forests. And not much was said about the role of the vast networks of mycelia in our farms and forests. And I was wondering to what extent those networks of mycelia contribute to forest and farm health and may be a stopgap against soil erosion.
They're hugely important. As you say, they do form these actual physical networks, and they also produce proteins that actually help soil aggregate, form clumps, and help it from eroding. So at many different levels, fungi are truly this network that helps tie things together. Once it gets to the point that you've got horrific erosion, it's very difficult. We hope to, not get there. But once you are there, there are ways that not only can you put down straw or different things, but people like Paul Stamets have looked at in his book, Mycelium Running, how you can put down straw and inoculate it with fungi in order to help tie things together and create a barrier that helps even more so with erosion, as well as helping to start that process of soil formation. And then we hope that in forests where you haven't lost the soil yet, to be able to promote things that keep the soil healthy and keep it intact, so that we don't have to be in that position of trying to start from step one again.
Yeah. Thank you. Go ahead.
How is climate change affecting fungus and its ability to decompose?
Okay. How does climate change affect fungus ability to decompose? Might be a couple of people on this. Amanda, you want, or I'm sorry. Yeah, Amanda, you want to give it a shot?
Yeah, just in the perspective of vegetable disease management, and I know there are others here on our panel that could address decomposition more perhaps specifically, but we do see we see changes in pathogen profiles, in terms of pathogens that used to be considered, at the start of my career, they were considered southeastern US pathogens of concern in vegetable crops, and now they're upper Midwestern pathogens that are affecting potatoes and pumpkins and various crops that we grow here. So there's certainly been a change in the species that are predominant. There's also a change in the intensity or the concerns with some of these pathogens, because our weather is more erratic. We have more moisture all at once in unique events. We have some warmer temperatures. We have more moderate winters. And so it's permitting the overwintering of these pathogens in our region here. But again, no, that doesn't answer the decomposition question specifically, but I thought I'd speak to that.
Yeah. Anybody else?
Yeah. Again, oh, sorry. Continue that question. No, I have an online question that.
Oh, okay. Go ahead.
Okay. Are there any other vegetables like corn that have an edible gourmet fungus associated with them, like wheat la coche? And are there any in Wisconsin?
We certainly have wheat la coche here in Wisconsin, right? It's interesting, depending on the corn type and the health of that crop, it can vary in colors and size and shape. But are there others? Across the vegetable crops that I work on, I can't think of another one. I can't think of another fungus that's pathogenic on vegetables that's a favored consumable.
All right. Do you have a question?
Yes, I do. Larry, I'm a big fan of yours on your radio show and all the other things you do. But my question today would be about harvestable mushrooms and what effect will the killing frost have? Is it applicable, or are there any types of mushrooms that can survive the first killing frost and that I could still be foraging in the woods for?
Ah. Well, I'm looking around here. Dan?
I'll jump in. The killing frost for plants is often a stimulation for some wood decay fungi. There are some fungi that actually need a good hard frost to get them to fruit. That one particular picture that I showed was of velvet stalks, Flammulina velutipes. It will come out, it loves a good hard frost and will often fruit after that. And sometimes you find it frozen. We wonder if it maybe disperses its spores during the winter, because you will sometimes find the mushrooms then frozen through the winter, and they'll thaw in spring. And so we transition from our summer foraging with a lot of the foliates and chanterelles, these mycorrhizal species, to many of the wood decay species, especially the oysters. Our native oysters that are still here will sometimes fruit before frost, and they're kind of white and thin. And on the same stump, the same one, after a frost, they'll come out much more robust and thicker with a browner cap. So it's not the end of the world that a frost is coming, but you will want to make sure you get your banana plants and other ornamentals inside. But many of the fungi are really good at overwintering and dealing with cold temperatures.
Thank you.
All right. Other questions? Someone else wondered, Dan, as long as you've been talking here, why did you wind up studying white nose syndrome in bats?
It comes down to DNA. One of the things is that with the Forest Service, we try and study the things that are most important for forest management. And it kind of came out of nowhere. And what it really came down to is fungi are really cryptic and hard to find and identify. Only 10% of them have been named and characterized. So when a disease like that comes along, be it chestnut blight, Dutch elm disease, white nose syndrome of bats, it's almost always an undescribed fungus, and we know little about it. And it comes down to DNA. I had just been working on how you could go into a log and pull out all of the DNA to look at all the fungi that were there and identify them so that you didn't have to rely on the mushrooms, because mushrooms are unreliable. Golden oysters might fruit, but when you don't see them fruiting, the golden oyster mushroom is still living in that tree. So they're cryptic and they're hard to find. And I spent a lot of time working with how to use DNA to detect things. And it just happened that the National Wildlife Health Center is in town here. They're an amazing group. David Blayhart and others were working on trying to figure out what was killing all of the bats. And we got this intersection between the wildlife people and people who study fungus who really hadn't intersected before. And that's how, at first it started out with just how can we detect it? What is this thing? What is this fungus? And how can we figure out where it is based on its DNA? And that's what drew me in. And then I spent also then the next 10 years on it, because I'm a sucker for any fascinating fungal story. And it's this fungus that has been killing bats is a very, very interesting story, and one that was really interesting to try and figure out how we could possibly come up with management measures for the fungus, as well as management techniques to keep our surviving bat populations alive and healthy.
Well, thank you. And on that note, I want to give a big thanks to our 5 experts today. It's been a lot of fun for me to listen to them and talk with them, and I hope it's been that way for you as well. Thank you very much.